Webinar number 4, Attracting Funding Opportunities and a Look Toward the Future of Ocular Drug Development. I'm Ashley Winslow. I'm CSO for Odylia Therapeutics, and we're a nonprofit biotech with the mission of accelerating therapeutic development for rare diseases. If you'd like to learn more about Odylia, please visit us at odylia.org or sign up for our newsletter. It's a pleasure to partner with Foundation Fighting Blindness on this educational webinar series. As I mentioned earlier, this is the 4th of our 4-part series. We're exploring new potential things to discuss in the future, so please keep tabs on social media outlets for both groups or our newsletters to hear more. And before I turn things over to Chad, who will be chairing our session today, I'd like to review some logistical details for the session. Currently, all participant lines are on listen-only mode. To ask questions after each talk or during the panel session, please use the chat icon or raise your hand. And just so you know where those are, chat icon is at the bottom of your Zoom screen. And then raise your hand is under the reactions icon, smiley face with a plus sign. You'll find raise your hand. So go ahead and press either of those. If you raise your hand, you will be called upon. You can turn that off if you change your mind. So also today's session will be recorded and will be available for viewing at a later time on Foundation Fighting Blindness YouTube page. With that, I'd like to introduce Chad Jackson, who's Director of Preclinical Translational Research at Foundation Fighting Blindness. The floor is yours, Chad.
Thank you, Ashley. And first and foremost, I'd like to give a big thank you to Odylia Therapeutics. This is our 4th webinar, as Ashley has mentioned, and this was sort of just an idea just about a year ago. And then we worked hard together to bring this to life. So thank you very much for being such a wonderful partner. The purpose of this series is to share tangible preclinical translational research and development information to the drug discovery community, focused on providing solutions to inherited retinal disease, as well as the broader rare disease field. We kicked off this webinar series back in October 2020 with our first webinar that was entitled Tools for Preclinical Development, creating a target product profile and effectively engaging regulatory entities. This was followed by a webinar in November called Disease-Relevant Translational Models in Ocular Drug Development, Types, Challenges, Advantages, and Alternatives. And this past January, we had our 3rd installment entitled Working with Contract Research Organizations and Contract Manufacturing Organizations to Accelerate Development. Each of these webinars were taped and are available for viewing on the Foundation Fighting Blindness' YouTube page. Today, we are here to discuss a very important topic, one that comes near and dear to my heart when it comes to really taking ideas from the bench into the clinic and the sort of orthogonal issues that surround that, mainly funding, as well as the sort of entrepreneurial spirit. So what does it take to push these technologies toward the clinical use and to the patient population that needs them most? So we'll sort of, from our speakers, understand a little bit more from their aspect. Excuse me. We will then wrap up the session with an open discussion so the audience has a chance to ask more questions about accessing funding from venture capital or from government programs such as the Small Business Innovation Research Program that will be talked about later, or just to have general views on what it's like to be an entrepreneur. We also give a look towards the future of ocular drug development and just get some people's general ideas or questions around what excites them most about what's coming down the preclinical pipeline, or just wild ideas that they would like to see tried to help mitigate some of these diseases. The foundation, as well as Odylia, we're very thankful to Doctors Denise Delcara, Rusty Kelly, and Payek Lee, who will lead us through today's event. Throughout today's event, we will actually have some polling questions because we don't want our audience out there just to be sitting back and relaxed. We want you to join in as much as possible for the discussion. These responses to the polling questions are anonymous. It's a great way to understand who's joining the event and get additional information as we step through the metrics. We kindly ask you to participate by answering the questions when they are presented. And actually with that, can I ask Leila, or Madison, could you actually do the first question, which is, what industry are you currently working in for the audience? So if you just take time to answer that poll, we'll give you about 30 seconds. Right now, academia is winning. Small business is next. Academia is way out in front. Yeah. We need more people starting businesses, Rusty. I see you have academia sort of leading the way out here. So great. If we go ahead and end the polling, I think we have about 55% from academia responding. That's awesome. 10% from industry, 15% in the startup small business realm, and then other was the other 20%. So great to know everybody out there. I have one more polling question before we kick off to the first speaker, and that is, have you ever started your own biotech business? Can we launch that? Just give it a few seconds here, maybe 15 seconds of responses. Great. So it looks like 86% have said no. So hopefully out of this webinar, you come away with at least an appreciation for some of the aspects of starting a biotech business. And maybe when you see a technology or something come across your path, you'll have the at least a little bit of the knowledge to go ahead and champion that and maybe start your own business. Thank you all for participating in that. Our first speaker, is actually going to be Denise Dalcara. She is a tenured researcher at INSERM in Paris, France, and leads a team on gene therapies and animal models of neurodegenerative disease at the Vision Institute in Paris. She graduated from the Middle East Technical University with a degree in biology in 2001, and afterwards she obtained a master's degree in pharmacology and pharmacochemistry in Strasbourg, France, where she later pursued a PhD in cellular and molecular aspects of biology. She was awarded the Bi-Valley PhD thesis for a method of protein delivery developed during her graduate studies. Later on, she conducted postdoctoral fellowship in the laboratory of Ernst Babmerg at the Max Planck Institute in biophysics before moving to UC Berkeley to do a second postdoctoral training in 2007. At UC Berkeley, Dr. Dalcara applied viral engineering principles to enhance AAV vectors for the application of retinal degenerative diseases. She has awards everywhere sort of down the gamut, and to read her entire sort of list of accomplishments would be too much here, but just know that she is an emerging leader in the field and is an active player in mitigating retinal degenerative disease. Dr. Dalcara, will let you take it away.
Thank you, Chad, for the kind introduction. So I'd like to start by sharing my screen. So I prepared a few slides to kind of tell you my story. Can you all see my screen? Okay, great. So thanks for coming to listen to this webinar about attracting funding opportunities and a look towards the future of ocular drug development. So the focus today, as Chad already mentioned, is taking a technology out of academia and putting it on a business path. And that goes through funding and entrepreneurship. So I'm going to give you my limited point of view on this experience of finding funding and entrepreneurship that has started for me not so long ago. But just to put it all in a context, I'd like to, first of all, give you a little bit of an overview of career path that I've had in order to sort of accumulate the knowledge and experience that sparked the interest in translational science, and that sparked interest then in entrepreneurship. So as Chad already mentioned, I've trained in nanotechnologies as a PhD, but then in postdoc, I've done work on optogenetics, which is also a new and emerging technology, and viral vector engineering. And these two have become really important in vision restoration and gene therapy. So basically, by going through these years of experiences, I think what was important was to get exposure to a lot of new and emerging technologies, because although as a student and as a postdoc, we don't really see it that way, the future is actually nearer than we think. So it's really important to get exposed to all of these new biotechnologies that are hot off and that are starting to make changes. And then with that experience, what is really important then when we become independent is to have a place to land so that we can leverage that experience and what we've learned through training and make it applicable. And in my aim has always been to be able to do something good, biomedically speaking, and to help patients. So where I saw myself after the training period was at the Paris Vision Institute. And landing there was really important for being able to then gain the vision to be able to go towards really translating the things that I've learned to meet the needs of the patients. So I think this is something, a piece of advice that is even before the step one of how to go about translating therapies into the clinic. It's to choose a place where you're actually confronted to clinicians, confronted to patients, and where there's a DNA of entrepreneurship and creating spin-off companies. If that experience and those capabilities of technology transfer and exposing you to what you need to know about being able to make a marketable product, then you're at the right place. Then you can really make things come together and happen. So for me, Paris Vision Institute has been really the important place where these factors came together on top of my training. And that, for me, I think set the basis for being able to leverage the funding and think about the follow-up steps. And then I'm going to try to break this down to you in different steps to sort of demystify the different things involved in entrepreneurialships. So the step one, I would say, in thinking about starting a spin-off company is to know true patient needs. And like I mentioned just before, for knowing true patient needs, you need to be where true patients are, and you need to work with people who know their needs and who are able to bring complementary expertise and points of views to your own in order to have a broad enough point of view and understanding of what true patient needs are. And of course, then these true patient needs are the underlying structure of the market need. So I think the step one really in starting all of this is to know these two things. And then you need to start with an idea that is going to correspond to a true patient need. And that idea is going to be developed probably in an academic setting with the help of the usual graduate students, infrastructures that are necessary to make this happen. But when you start rolling this machine, it's important also, I think, for people with application in mind to be sure to look beyond the rodent model. So on the FFB website, in past webinar series, I've seen really wonderful webinars that are still out there about different models. So I encourage everybody to check these out, because we're really in a stage where, especially in the gene and cell therapy fields, there's a lot of stuff that is in the pipeline now and that is wanting to reach the clinic. And I think what people who are the funders, the stakeholders in this, are really focusing on experience that looks beyond just rodents. Of course, even in looking at creating patents, I think the ability for a tech office to really take up an idea and want to start the process of putting a patent on it is going to depend on first generating in vivo data. And then the in vivo data, you should really by no means stop at the mouse or rat model, because then you're just going to be optimizing your technology for the wrong person. No matter what you do to a mouse, he's not going to have 20/20 vision. So I think it's really important to keep that perspective in mind when you have when you're working towards something that can be applicable. So that being said, so that's the first step. Know the true patient needs, true market need, and create IP. So that's the step one, I think, of any entrepreneurial venture. And then the step two is, of course, to attract partnership and seeding funds. So without funds, none of this can happen, especially in the field of emerging therapies that we have today, where gene and cell therapy are really big players. These are expensive technologies, and attracting partnerships and seeding funds is absolutely key. So for me, in my experience, this has been done thanks to Advent Trans Biotechnology, who actually noticed my work and who approached me. And this is kind of when I thought to myself that I have an idea, maybe I have an idea that is worth bringing into the clinic. And I was also thankful to them bringing in a qualified and experienced team that complemented my scientific expertise with operational and business experience. And they already had an established track record, so it was really easy to to form a partnership with them and to start this collaboration. And they leverage funds from the European Investment Fund and the European Commission, and they have really tremendous experience in taking these early stage and under, let's say, under the radar kind of projects, and you know, going to academia and showing interest in these projects, and then growing them slowly into with a first seed round, and then attracting additional funding via their management team, and having them grow eventually towards bigger companies. So step 2, attract partnership and seeding funds. And I think that's going to also give you the infrastructure of the legal and administrative aspects of implementing a company, starting a company, and all of these things that you don't know as an ordinary academic scientist. So hopefully that kind of partnership can really come in handy also for those aspects. So that's step 2. And step 3, then, once you've implemented the company, attracted the funding, is to surround yourself with a network of collaborators, which is really essential to the project. So again, I'm going to take it back to the beginning of my talk. I think being here in the Paris Vision Institute was really important for that, because all of the, almost all of the founders, co-founders of Gamma Therapeutics, my company, they were already clinicians or scientists working in this institute, and working with them and collaborating with them has been just more than natural and easy. And I really benefited from their complementary expertise, which not only allowed me to have a clear vision of the clinic, but also in terms of going towards follow-up product ideas and follow-up developments, which is really important in establishing the company. So all of these worked very smoothly thanks to the identified founders and also the key personnel. So the first 2 recruits that we've done in the spin-off company were my previous graduate students, which I knew were at the basis of the IP that attracted the seed funding and that attracted the attention to the spin-off. So having them has been really important in being confident in being able to go on about the next steps, which are to incorporating the company and establishing a development plan. So once you have attracted the funding and you've established a network of collaborators and founders of the company, next thing is to come up with a development plan. A development plan is basically that little patch that is going to bridge the gap between everything you know from preclinical studies to the clinic. So it's going to be those essential steps that are the lingering concerns about the translatability of your project and how what you found in the animals is going to be applicable to human beings. So what is missing is going to be the question in coming up with milestones and deliverables that are going to then eventually de-risk the project and make it as little risk as possible to then finally go on and take that step towards clinical application. So step 4, incorporate your company and establish a solid development plan. And while establishing that plan, get plenty of people on board and who have experience with this type of plan. So you have goals that are realistic, that de-risk the path, but that don't create supplementary obstacles, especially in terms of the regulatory issues that are going to be posed afterwards. And then once you've come up to there, you have a plan, you've raised funds, you have seed capital, you're ready to advance your technology or technologies through successive product development milestones. And there, time is going to be a very big issue. I think something we are very concerned with is checking these deliverables and milestones on time, finding reasonable replacement strategies, and basically not getting blocked at any go, no-go steps. Those are going to be key in successfully developing your product. So, and I think after the 5 steps, once all of that is accomplished, I think for an academic researcher who keeps their daytime job, which I advise everybody to do, it's going to be really important is to take this experience and bring it back to the lab So you can have a next line of products that can be developed from what you learned from bringing this product all the way to the patient. That there you've seen the needs, you've seen what is met by your product and what is still not met, and how that can give rise to the next next horizons, basically, in terms of making even more relevant therapies. So with that, I think I've said everything about my experience. And here's a little glimpse into Gamut Therapeutics. So you see the people and faces associated with the project. So the scientific founders are myself, along with Stefan Hurlitz from the Bochum University, Olivier Gouraud, Isabelle Otto, and Serge Picot, who are all from the Paris Vision Institute. And we're extremely fortunate to have a very good management and advisory team who are complementing us. And we are very happy to be working on our current project on gene therapy and cell therapy approach to restore vision in late stage retinitis pigmentosa patients. So with that, thank you all for listening.
Oh, Dr. Dalkar, thank you so much for such a great presentation. I think it definitely gives us and the audience a great foundation for those who are interested in sort of starting their entrepreneurial journey. So thank you for that. I would like to let the room know that if you have a question, either please raise your hand and we'll call on you. You can unmute yourself and ask your question, or just type it in the chat box and we can ask from there. I'll just start off real quick just to get the questions rolling. You sort of initially talked about true patient needs as far as when you're thinking about your particular technology. At what stage in your research are you thinking about that, and how are you sort of obtaining that sort of true patient need? Are you interacting with clinicians, patient advocacy groups? How did you approach it?
All of the above, actually. I think I thought in my mind when I was a postdoc in Berkeley, I thought I was doing really applied research, and I thought I had patient needs in mind, but I was far from understanding that my mice were not really patients. And all of that realization of this gap between the patient need and the mouse need really became much more evident to me once I came here and I started interacting with patient advocacy groups and other researchers and clinicians who were really by the side of the patient. They kind of gave me a wake-up call. They said, hey, Denise, you really have no idea what is going on this side of things. And I think the word translational research, the way José Sahel, he taught me to understand it, it also is a language issue, really. Translational science does not just mean translating experiments from bench to bedside, but it also means translating different languages spoken between patients, academicians, industrials, and clinicians. These people don't speak the same language and they don't understand the same things. When you spit out a sentence, it doesn't mean the same thing to them. So learning those different languages, understanding them, and being able to speak to each stakeholder is really important in understanding the so-called patient need.
Great, thank you. Yeah, follow-up question to that. Can you give us a little bit of insight of sort of the beginning of your journey when the sort of the group that you mentioned reached out to you about potentially, sort of investing in this particular in your technology? What were some of the either emotions or first thoughts that you had, when they approached you, that I'm sure many people who have never sort of taken this journey might have at the onset, and if there were sort of some hurdles there, could you explain maybe how you got past those?
So honestly, maybe my experience is a little bit unique because, like I said, here we have a tradition of creating spin-off companies and very active exchanges between industry and our institute. So I was already bathing in all of this when Advent approached me, and to me, maybe I was a little bit blasé. I mean, I didn't really think it was that big of a deal. I was like, because prior to that, I had discussed with many different entities, and there had been projects that didn't really then follow on. So I thought, oh, maybe it's another investor checking things out, but I didn't really see it like they were really going to commit. So at first it was kind of flat, but then as we went on meeting more and more, I started understanding that their interest was serious, and they also started being convinced about the potential of the technology. So there I thought, oh, well, I really have to decide now if I want to be part of the adventure or if we just license the stuff and I stay in my academia. So then it was kind of a crossroads, and maybe that's where there was a little bit of emotion. I said, okay, maybe I should. It is my duty also to accompany these technologies so that they can do their best in finding their way to application.
One thing you mentioned that was very interesting to me is the environment. And one of the issues that I worked on for a number of years is sort of what we call maybe innovation ecosystem building. But could you speak just a little bit more about the environment, about being around sort of being in an environment that really champions entrepreneurship as well as the startup, sort of the startup arena, versus maybe being somewhere that doesn't have that particular resources at hand? Because I'm sure that on this call we have many people who come from many different environments and may not have the resources that you have. Maybe you could just give them a little bit of insights to some tools they could use to maybe help them along their journey.
Yeah, unfortunately, this kind of thing, you can't really create it out of thin air. I think I really was privileged to have this opportunity because this is the vision that José Serral created by establishing this institute in the parking lot of the 15 Eye Hospital. This was his vision for what he wanted the institute to become. It was really to implement, to make this triad between the industry, the researcher, and the patient, so that this kind of circle would start around discovery, application, and coming back to the starting point. So I really benefited from years and years of having worked on that vision and having established something that corresponds to that vision. I just came and just was able to operate that system. So I think for somebody who doesn't have that context, it might be a little bit difficult to create it out of just a purely academic environment. But that being said, with the way that our institute was established, I think it's inspired a lot of other people to think about this configuration. Now, a lot of people are following this idea. For example, Kobe Eye Institute, created by Masayo Takahashi, she has kind of a similar vision in mind now in creating this kind of environment for her researchers. Same with the Tübingen Eye Center is kind of established around a similar idea where neuroscience really meets the needs of patients. Same with the Institute of Ophthalmology of Basel, which is already thriving from tight connections between industry and academia, and it's really also being founded on this kind of idea. So I think now this concept is really is growing, and I think people might find opportunities in different places in the world of finding themselves a context like this one. But I think it would be extremely difficult to create it out of nothing in an obscure university with just an academic environment.
All right, thank you so much. And again, thank you for your presentation and your insights. I greatly appreciate it. And we'll have you hold on a bit until we get done with the other two speakers and come back for the open discussion. So thank you again. We do have another polling question before we move on to the next speaker. Madison, could you please put the third polling question up for the group? And it is sort of just a fun question here. What round of funding, best defined by the investment, used to take a startup company from the idea to the first steps, such as product development or market research? The audience, if y'all please could. All right, thank you. So we have, all right, Rusty, how do we do? We got 23% saying Series A and 77% saying seed round.
Seed makes the most sense.
There we go, seed round. Seed round is correct. Thank you all for participating again. We appreciate it. Our next speaker is Dr. Rusty Kelly, who is the Senior Vice President of Investments and Alliances at the Foundation Fighting Blindness. He is responsible for the external investment opportunities that strategically align the foundation's therapeutic mission and its venture arm. This venture arm is called the Retinal Degeneration Fund, or RD Fund for short. Prior to joining the foundation, Rusty led the interdisciplinary and translational funds at the Burroughs Wellcome Fund. Prior to Burroughs Wellcome, Rusty was the head of preclinical at TGen, a tissue engineering biotech out of Boston Children's and Wake Forest University. Early in his career, Rusty was under multiple divisions in clinical development at PPD. Prior to PPD, Rusty was on the bench in the pharmaceuticals and analytical chemistry divisions at AI Pharma. Rusty received a BA in chemistry from UNC Chapel Hill, a PhD in pharmacology at LSU's Health Science Center, and was an American Association postdoctoral fellowship in the School of Medicine at UNC Chapel Hill, and has recently earned an MBA from Duke University. He is my trusted colleague who leads us through the navigating the water of really sort of putting the gas behind the translational vehicle at the foundation. So with that said, Rusty, I'll turn it over to you for your remarks.
Great, thank you, Chad. It's a pleasure to be here with you all today. And thanks to the Odylia team, Ashley Winslow. We really appreciate this partnership. So I think I'm going to reinforce a number of the points that Denise has already made. Really appreciated her talk. I think it's fundamental, this triad that Denise speaks of between academia, the patient, and industry. And I think the foundation really sits at the center of that triad. We aim to serve all three aspects in different ways and in multiple ways. But as Chad mentioned, my background is varied with experience in biotech, including on the bench, as well as my education in chemistry and pharmacology and business school. I've really migrated to the business side of things, and that's for a variety of reasons, but most importantly, when you're in biotech and you're on the bench, and you're not necessarily in control of management decisions, things can go south or awry very quickly. It's a difficult business, and I think Denise and my colleagues would attest to that. There are lots of reasons for failures, and it's these reasons for failures that are also become the reasons for criteria, for investment criteria. The whole goal is to de-risk. So you have to identify those risks, and you need to figure out how to fill the voids, fill the gaps. If it's management, if it's the technology, it can be a variety of reasons, including your financing strategy. But I'll tell you a little bit more about the RD fund. So the RD fund is the venture arm of the foundation, and I came to the foundation because Ben Yerxa and the brain trust of the foundation, an existing management team, decided that it would be prudent for the foundation to create a venture arm to serve not only the mission of the foundation, which we'll talk about, but to also provide an alternate source of income. So the RD Fund is a 501c3 not-for-profit subsidiary of the foundation. Let me make sure I'm sharing my slides here. Chad, I failed to share my slides, so let me do that real quick.
No problem, take your time.
Pardon me for 1 second. Let's see here. Having some difficulties here. Share screen, desktop 2. Can you see my screen now?
We can see it. It's still in, it's not in presentation mode just yet.
Okay.
But you probably could work off of that if you'd like.
I'll just work. Yeah, I'll just work off that.
Okay, great.
Okay, so let me move to slide 2 here as we move into the overview and eventually the investment thesis. So as I was saying, the RD Fund is a 501c3 not-for-profit subsidiary of the Foundation Fighting Blindness. The foundation is the sole member, and therefore there are no limited partners or LPs that invest in the fund, as you would see with a traditional venture capital firm. All of the funds under management are donated. It's very unique. This is a first of kind fund that focuses only on inherited retinal degenerative diseases, and it's an internal dedicated venture arm. So venture philanthropy is this term that's used quite a bit, but this is a type of impact investment that takes concepts and techniques from venture capital finance and business management and applies them to achieving a philanthropic goal. In our case, this is via mission-related investments that aims to provide both clinical and financial returns, as I mentioned earlier. The fund was launched in late 2018 with 72 million under management, and all proceeds that the RD Fund receives back over time from these investments are returned to the foundation. And it's up to the foundation to determine how those funds are used, including to support further research and or to fund the RD Fund in its subsequent funds. So we are a closed-ended fund, 10-year fund. We are in the final stages of spinning out Fund 1, and we are fundraising for Fund 2. So in terms of exit and returns, for example, the fund would receive funds back when a portfolio company is sold or becomes publicly traded. In fact, we just had our first exit in October of 2020. Videre Bio was acquired by Novartis for $280 million. The foundation is currently fundraising for major gift, or I should say major gift donations for Fund 2. So we're approximately 90% committed for Fund 1, and Fund 2 is certainly reeling off of the success of the Videre exit. So slide 3 talks about the thesis of the fund. So most funds will have a thesis, and that thesis can evolve over time. Ours certainly has. But our thesis in general is that we are geographically agnostic. We are funding or investing all over the world, including in Paris. It leverages the capital of other major investors. And in fact, we are approximately 5.5-fold greater than RD Fund investments from outside capital among our current portfolio of 10 companies I'll tell you more about. As I said earlier, we're investing primarily in therapeutics companies, but we're also open to other technologies that support the foundation's mission. We focus primarily on companies with programs that are in clinical testing that can be in the clinic in less than 18 to 24 months. Our investment allocation ranges between 2 and 5 million with appropriate reserves to provide each company additional funding as needed. And we use a variety of investment strategies, including convertible debt, equity, royalties, and or non-dilutive, as they would say, project-based co-funding or non-equity-based funding. The RD Fund leverages the breadth and depth of the foundation's knowledge, global relationships, and resources, including its clinical consortium, My Retinal Tracker Registry, our scientific advisory board, and again, the outside funding from our co-investment partners. So slide 4 is a simple statement of our mission. As I mentioned earlier, we are focused purely on inherited retinal dystrophies. And it's really about finding preventions, treatments, and cures for our patients and our constituent groups. So slide 5 talks about the governance of the RD Fund. So we have an independent board of directors with representatives from the Foundation Fighting Blindness and other experienced community members who bring significant scientific, clinical, and financial expertise to the team. Warren Thaler is our RD Fund board chair, and Warren is a longtime key supporter of the foundation and has served as the foundation board member for many years. Warren is joined by David Brent, chairman of the foundation, Dr. Jackie Duncan, UCSF ophthalmologist and chair of the foundation's scientific advisory board, Dr. Jonathan Steinberg, chair of the foundation's research oversight committee, Dr. Adrian Graves, former CEO of Santen, Kelly Lisbakken, managing director and head of biopharma investment banking at Wedbush Patgrau, and Dr. Jean Dewan, vice chair of Foresight Labs, a serial inventor, entrepreneur, and ophthalmologist at UCSF as well. And importantly, one of the foundation's founders and RD Fund anchor donors is Mr. Gordon Gunn, an honorary and active board member. And then slide 6 briefly covers the management team of the RD Fund. And you can see that Ben Yerxa has recruited a team of individuals that have significant biotech experience. And that's experience across the board, both scientific, business, as well as on the marketing side. Jason Menzo. Slide 7 is a schematic that shows our portfolio. So as I mentioned, we've invested in 10 companies thus far. And these companies, as I mentioned, include globally represented companies or multinational companies. And they also include internally conceived startup companies working on a range of promising technologies and therapeutic targets, including gene therapy, RNA therapies, pharmacotherapy or neuroprotection, optogenetics, and digital technologies. So these 10 companies at Athena Therapeutics, Checked Up, Lookout Therapeutics, Nacuity, Nyan, Procure, Sparing Vision, Stargazer Pharmaceuticals, Videri, and then its spinoff, Videri Bio2, following acquisition by Novartis, represent the 10 companies that I mentioned earlier, with approximately 50 million currently committed capital and with approximately 15 million in capital reserves. And this, again, this commitment represents about 92% of our 72 million under management for Fund 1. And I think importantly, Denise indicated the importance of smart founders, experienced founders, but this is also true of your board of directors and those that invest in these respective companies and take these board seats. It's very important to have good board dynamics. And this slide shows you the top tier firms and strategic partners that we're surrounded by in this current portfolio. So we're off to a very good start in creating a very strong ecosystem of investors around the foundation and the RD Fund. So a couple more slides. I think the strategy for the RD Fund is extremely unique, and it's been well received by our co-investors. So our model that is hyper-focused on IRDs takes advantage of a half-century of funding research by the foundation. And I can't emphasize the importance of this enough. The foundation has a strong reputation for strong science, and we, the RD Fund, are really taking advantage of that reputation. And so it really starts with this concept of high science or smart science. So deep insights regarding therapeutic targets, genetics and modalities. We have significant deal flow, including from the foundation's ongoing grant support. We have all the foundation's resources that we discussed earlier, including this registry that's very powerful. We have a flexible investment strategy, including the freedom to make both equity and non-dilutive or non-equity based investments. We're well connected to an extensive SAB and KOL network for diligence, as well as their participation on respective companies' scientific advisory boards. So in summary, the success of this model is based on leveraging our funding with other investment firms and strategic partners that allow us to accelerate more opportunities. So we can take some of these threads into Q&A, but I'm happy to tell you a little bit more about our selection criteria for companies during the Q&A session. So thank you for your attention.
All right, thank you, Rusty, so much for the slides, for the information. I think they're very important, especially for those who have not worked with a fund before and are looking to potentially access that type of a support in the future. I think we probably have time for a couple of questions. So again, from the audience, if you have a question, either type in the chat or raise your hand and unmute yourself, and we'll definitely let you speak to the panelists. I'll just start it off. And speaking to a little bit of that, Rusty, what would you say are some of the important things for people in academia to consider before approaching a venture capital firm, as far as the information they should have ready, or some of the questions they should have answered before going into that particular meeting?
That's a great question. I think Denise indicated all the aspects of company creation and the general guidelines for what constitutes a healthy startup company, for example. So there's the science, of course. Everything's founded on good science, and whether that science is suitable for technology. So I think there's a reputational aspect of that, and again, the foundation's had the advantage of investing in individuals that we've granted towards over the years. So there's a strong relationship with these individuals, and there's a strong understanding of that science. But I think it's really about packaging that up correctly. So the team is important. Denise indicated that. The commercial opportunity is obviously the bigger the market, the better. Your financing needs and your financial position is certainly critical. But I think since the majority of this audience is from academia, it's about packaging that science and that ability of that science to translate into technology up appropriately. So what is the important data? What are the gaps in that data? You're not expected to have everything that would be suitable for a clinical trial, for example. So we need to understand what is the research stage of this candidate product, and then what preclinical studies need to be laid out that enable that technology and to make that technology investable. And that also includes the intellectual property position and your ability to sort of operate, the freedom to operate, if you will, in a field that especially a field that may be becoming fairly competitive.
All right, thank you for that, Rusty. And I'm not seeing any questions you shared from the audience, but I think we'll just move on right now and save some of the other questions for the open discussion. And so thank you again, Rusty, for that. And just hold on and we'll be back with you shortly. Before we move on to the next speaker, we have another polling question. So Madison, if you wouldn't mind putting that up there for the group. And this is, how many federal agencies participate in the SBIR program? While y'all are taking your time, it's either 10, 11, 14, or 12. It's a very hard question, I guess, unless you just Google it, because I had to Google it when I was making this polling question. So, all right, so I think we have it close to the poll. We have 38%. Somebody revoted. All right, so the answer is actually 12. There's 12 federal agencies participating in the SBIR program. And we're lucky enough to have with us today Dr. Peg Lee from the National Eye Institute, or one of the agencies actually involved in this program. Dr. Lee is the program director for NEI's Small Business Innovation Research and small business technology transfer research portfolios, and also serves as the NEI's Research Resource Officer. Prior to joining NEI, Dr. Lee served as a referral officer and a scientific review officer at the Center for Scientific Review at the National Institutes of Health. As a SRO, he managed numerous NIH peer review panels, including the Aging Development, Auditory, Vision, and Low Vision Technology Small Business Panel, and the Molecular Neurogenetics Study Section. Before joining CSR in 2003, he was an intramural researcher at the National Institutes of Neurological Disorders and Stroke, and he has a PhD in neuroscience from Kent State University. His research experience was in the field of neuroscience, ranging from neurotrophic signaling to neuroimmune mechanisms underlying neurodegenerative conditions such as multiple sclerosis and diabetic neuropathy. Dr. Lee, the floor is yours.
I want to thank Chad for inviting me to this webinar. Hi, everyone. My name is Pak Lee, Program Director for NEI Small Business Research Program. And I would like to share my screen at this moment. Can everyone see my screen?
Yes, we can.
Great. So during my talk, I would like to close my screen off so that people can pay attention to my talk. But I guess I won't be able to do that. So I'll leave it up to you. If you'd like to reach out to me with specific questions with regard to your own project after this session, my email address is listed on this slide. Our small business grants management specialist, Ms. Leslie West-Bushby, is also on this meeting, and she will back me up with the technical details of a grant management process. So Leslie, you may introduce yourself.
Welcome, everybody, and good afternoon. Yes, my name is Leslie West-Bushby, and currently I work for the NEI. However, I have a background as working for a private biotech company. So I have about 20 years total between the private sector and the government sector, 5 being in the government. And so I have the advantage of knowing both perspectives. And so hopefully I can answer any questions that you may have.
Thank you, Leslie. So today I'm going to talk to you about NEI's Small Business SBIR STTR program. And I have 3 goals to accomplish as shown on this next slide. First, I will introduce you to NEI's extramural research programs and priorities. For those of you who are not familiar with what we do in our extramural divisions, then I will go over a high-level overview of our SBIR and STTR program. And then I will walk you through the grant funding process that it takes for us to fund an SBIR or STTR grant application. So NIH's mission is described on this slide. NIH's mission is to seek fundamental knowledge about the nature and behavior of living systems and the application of that knowledge to enhance health, lengthen life, and reduce the burdens of illness and disability. Our NEI mission is pretty much in line with NIH's mission, but focused more on eye and vision-related disorders and knowledge about visual functions. More specifically, we provide financial support for research, training, health information dissemination, and other programs with respect to blinding eye diseases, visual disorders, mechanisms of visual function, preservation of a sight, the special health problems and requirements of individual with impaired vision, as well as providing eye health care to underserved populations. The good majority of our funding goes to nonprofit research institution, but also a small but significant portion goes to small businesses for the express and accelerate the purpose of a commercialization. We also support translational research that focuses on turning important knowledge into useful technology and products, which is also called discovery to clinic or discovery to market. With that, our extramural research programs currently include the following research programs and priorities based on anatomy-based and disease-based areas. They are retinal diseases, corneal diseases, lens and cataract, glaucoma and optic neuropathies, strabismus, amblyopia, and visual processing or SAVP, and low vision and blindness rehabilitation, as listed on our internet site. In addition to these anatomy and disease-based research programs, we have cross-cutting programs as listed on this slide, and the SBIR STTR program is one of them. In addition to that, we have a collaborative clinical research or UO1 mechanism, Center Core Research Program, Translational Research Program TRP on therapy for visual disorders or R24 mechanism, Conference Grant mechanism or R13 mechanism, and training and career development for F31, 32 fellowships and T32 training grants. For more details of these programs, once again, you can check out NEI's internet site, or if needed, please reach out to individual program directors listed on our website. So now NIH Small Business Research Program is limited to domestic US-owned and US-operated small business concerns or SBCs, and it is a congressionally mandated program. It was specifically designed to stimulate technological innovation to meet federal R&D needs and to increase the private sector through federal R&D funding. The objectives also include fostering and encouraging entrepreneurship by socially and economically disadvantaged SBCs and women-owned or women-run SBCs and SBCs operating in underrepresented idea states. So our program can be divided into two distinct mechanisms. Those are Small Business Innovation Research or SBIR and Small Business Technology Transfer or STTR. The SBIR is a set-aside program for SBCs that have research projects that have a high commercial potential to engage in the federal R&D effort. So all federal agencies with extramural research budget over $100 million must set aside 3.2% of their annual budget for SBIR program. Its phase 1 mechanism is called R43 and phase 2 R44. It currently utilizes approximately $22 million from the NEI annual budget, and it is specifically requires a 2/3 of research effort performed by the applicant SBC, and 1/3 can be outsourced to a collaborator or subcontractors. On the other hand, STTR is designed to facilitate cooperative R&D between the applicant SBC and US research nonprofit institution, including academic labs. When a tech with a high commercial potential is existing between them, you are a good candidate for an STTR application. Phase 1 of these is called R41, phase 2, R42, and it currently utilizes about 0.45% of NEI's annual budget, or approximately $3 million. It requires at least 40% effort performed by applicant SBC and 30% by the academic or nonprofit collaborators And the remaining 30% can be done by either of these parties or a third party. This slide will show you 3 different phases a successful SBIR or STTR project may have to go through. There are phase 1 discovery phase, phase 2 development phase, phase 3 commercialization phase. So our program prioritize its investment mostly into phases 1 and 2, and the phase 3 steps are largely outside of our funding priorities. So phase 1 projects are mostly feasibility or proof of concept testing studies, and the research plan should not exceed 6 pages, and the budget should not exceed this amount listed here, which is updated every year. Right now, it's at $256,582 total cost for the duration of 6 months to 1 year. For phase 2, an additional section called a commercialization plan is required. The research plan alone can be as long as 12 pages. The budget cap for this mechanism is currently at $1.71 million for the total duration of 2 or 3 years. The budget exceeding these caps can be justified for a set of waiver topics NEI currently has, but you would need to check with me prior to a grant application submission. More details of the NEI waiver topics, you can check out the program description document available in NIH SBIR website. For phase 2, we encourage you to reach out to private funding, and you can combine federal and private funding. For phase 3, as I said, it should be done by using non-SBIR funds, and you may primarily rely on private investors for your financial support and FDA to navigate through the regulatory path to bring the product on the marketplace. So this slide shows you different options that you have with our program. The small business application types that NEI supports are listed on this slide. A normal sequence of a project development would be to have a phase 1 done and then apply for a phase 2 grant. If the applicant finds it fit, a direct phase 2 option or a fast track option can be done. Direct phase 2 is for a project that has a significant phase 1 equivalent data using non-federal funds and thus are justified to skip the phase 1 part. The fast track option is for a project that is still in early stage but has a strong chance of success in their phase 1 portion, so that the applicant can submit a phase 1 plan and phase 2 plan together in one application. If a fast track is funded, the phase 1 is required to submit, the PI is required to submit their phase 1 progress report directly to NEI for our internal review after completion of phase 1 work, and the phase 2 portion of the grant will be approved if determined that all phase 1 milestones and specific aims are successfully met. We also have phase 2B and CRP mechanisms for super high impact projects that still have some research work and regulatory tasks left to be done. And these are rare in our portfolio. But you can go ahead and check with me before you submit your phase 2B SBIR or CRP application. None of these 3 options, direct to phase 2, phase 2B, or CRP, are available under our STTR program. This table inserted here should give you a pretty good idea about different options you can have with our program. Like I said, SBIR have all these options, including fast track, whereas STTR has R41 or R42, depending on your level of research development. So NIH grant funding process involves multiple NIH IC and can take up to 9 months. Understanding the process, including these elements, would help you prepare a good grant application. You would want to get familiarize yourself ahead of time with what types of electronic registration are required prior to a grant application submission. What specific FOA you are going to respond to is also important so that you will be able to prepare your grant application according to that specific FOA. Once you submit your application through the grants.gov website, the application will be assigned to a small business review panel that will produce a priority score between 10 and 90 and a summary statement for your application. Based on these review outcomes, NEI then will carry out pre and post award processes. Let us now take one step back and look into what electronic registration you would need to do to be eligible as an applicant SBC. Your company, not yourself, your company would need to register with all of these organizations. First, DUNS, to get a DUNS number, then EIN number for the tax purpose, and System for Award Management SAM number. With this DUN, EIN, and SAM number, you can apply for ARA Commons account for your organization, and the company should be able to designate a PDPI or a PI who will also need to have his or her own investigator account. You will also need a grants.gov account, as well as SBA company registry. And then you use grants.gov assist or grants.gov workspace to submit your grant applications. It may sound overwhelming, but if you plan ahead and take care of them one by one, it should be a pretty straightforward process, because some would take up to 2 weeks or long. We ask you to have a timely planning of these registrations, and it would be important to you make sure that these done ahead of time. When all these registrations are done, then you are ready to submit your grant application. To be eligible as an SBC applicant, the SBC has to be physically located in the US, and more than 51% should be owned by a US citizen or a permanent resident. The SBC with more than 500 employees will not be eligible for an SBIR application. The PI of an application can be a foreign national as long as he or she is legally residing in the United States. We have a set of omnibus funding opportunity announcements or FOAs. For SBIR, omnibus parent FOAs are divided into two based on presence of clinical trials in your studies. And 3 active CRP FOAs are also available for SBIRs, depending on the nature of the tasks remaining for your project. For STTR, omnibus present FOAs are also specified based on the presence of clinical trials. And all these details is available at NIH SBIR website. Those of you who are not familiar with what needs to be included in your small business grant application, you can check out these websites and resources available there. I recommend that you visit the 3rd bullet for the sample applications and summary statements especially if you are new to an NIH grant process. Your goal is to convince your reviewers on the review panel that you have a project that has an excellent chance of success and a high commercialization potential. We work with 3 independent review cycles based on 3 standard submission deadlines. So this slide illustrates the timelines associated with the 3 submission dates. For example, if you submit an application by January 5th, then the review panel will convene in late February and early March. NEI Council will meet in May or June to approve the final funding decisions, and the award may go out as early as in July. Of course, these standard dates will be affected by holidays and weekend. So whichever comes as a working day, business day will be the first deadline that you can use to submit your grant application. So what about the review process? Like I said earlier, the first level review will be carried out by a peer review panel within the NIH's Center for Scientific Review. The panel is composed of outside reviewers, non-NIH employees who have adequate expertise to review your application. And the panel is organized and managed by an NIH official called Scientific Review Officer, or SRO. What they evaluate is the scientific and technological merits of your application, as well as its potential impact. There are 5 scoreable review criteria. They are significance, investigators, innovation, approach, and environment. In addition to these, reviewers are going to be asked to evaluate the commercial potential of the SBIR application. So when I was a scientific reviews officer at CSR, I always asked my reviewers to start with 2 questions before they delve into 5 core criteria. First, should this project be done? Then second, can this project be done successfully? So if you ask yourself these 2 questions and your answer is not strong yeses for both questions, then you may have to go back to the drawing board and write up the grant application again. So this slide will give you a chance to look at a bit further into course review criteria. So if I walk down the list, the first will be significance, and significance is pertaining to the question such as, does the product address an important problem and have a commercial potential? Is there a market pull for the proposed product? More or less belongs to the first question that I asked, should this project be done? Then investigators, the investigators, collaborators, and their key personnel are adequately trained and capable of completing all project tasks. Innovation is not as important as other review criteria for small business grant applications, because a lot of small business applications do use existing technology. However, still the review will be looking into how novel the technology product will be, and then what types of application that you're going to use or indication of use are innovative. Approach, design and methods well developed and appropriate, problem areas addressed, potential pitfalls and alternative approach provided, is sufficient scientific rigor reflected in the research plan. And then they're asked to look into the settings that you're gonna do your project. So small business grants environment is key, because unlike nonprofit organization and academic lab, you have to establish your own facility resource and independency. And if you don't have that, the reviewers may not be really excited about your chance of success. And then lastly, commercialization is the company's business strategy, one that has a high potential for success. Finally, on the next two slides, I'm going to briefly talk about pre and post award process. These are very complex process and vary a great deal based on your individual situations. But what I will go through today is a standard version of it. Once the review meeting has been done and your score and summary statement that contains 3 to 4 critiques have been released, then your application will fall into one of two categories: to be funded or not to be funded. If your application was either not discussed or received a very poor score, unfortunately, then it will not be considered for funding. As you may know, NEI Small Business Program is a highly competitive program, and so our success rate is never high enough. Consequently, you would have to revise and resubmit the application, including one-page introduction where you will put in your response to the previous review if you fall into this category. If you're not sure what exactly needs to be done, you can reach out to your program officer. That will be me for NEI. If you received a highly favorable review and a very competitive score, then you can contact me too to ask what further step needs to be taken. Our grants management office will demand just-in-time information, but just because you received an email demanding just-in-time does not mean that you will get the funding. ERA sends out an auto-generated email, and our office also sends such an email to a large group of applicants who may or may not going to be selected for funding. So it is better for you to check out with me. If every requirements are met and your project meets NEI program priorities better than others, then you will receive a noticeable award, which means that you finally have the funds needed to start your project. The funding period starts with your receiving the notice of award. And we ask you to read it carefully. The funds will be available in the payment management system, from which you will be able to use the funds available there to carry out your research. Then we ask you to keep us updated how well you are progressing with the project. And once you have completed the project, then you are required to submit a final progress report and other closeout documents. Then if necessary, we will ask you further details. And then if you need to resubmit the grant application, you can start this cycle all over again. If you are ready to move on to phase 3 commercialization process, then you will be on your own, but we still want you to touch base with us and check back when you have some good news or breakthrough to share with us, as we will closely monitor your accomplishment. So to summarize, today I talked about the purpose of NEI Small Business Research Grant Program and what types of research efforts you should submit as a grant application, and what types of grants we support, and what are some of the important elements that we will look into determine your success in the first level peer review process, and some key steps needed in the grant funding process with NEI. These are some of the useful resources you can check out. And if you have a specific project to talk to me about one-on-one, you can always feel free to shoot me an email, and a process will start from there. I'm happy to answer any question you have, but for the moment, I will turn it back over to Chad. Thank you for your attention.
All right, Dr. Lee, thank you so much for that information. It's very important and very detailed. We really appreciate it. Again, I'll remind the audience that you can either raise your hand and unmute yourself to ask a question directly to our panelists, or you can type it into the chat box and we'll ask it for you. Dr. Lee, I just wanted to sort of start with just a general question, because you touched on a little bit about sort of who is eligible for it, and you talked about foreign nationals who are eligible if they reside within the United States. So my assumption is that for people who reside outside of the United States, they are not eligible. Is that correct?
Right. So there are two different eligibilities. One's for the organization or the company, SBC, and the other is the PI. And if the PI is physically located in the United States, is required, they can make travel and they can be outside temporarily, but their permanent residency has to be established in the United States.
All right, wonderful. The other question I had was, once someone is successful at obtaining an SBIR, is there anything that they have to be careful of those funds being in jeopardy, of sort of the NIH either pulling those funds back, or if the project's not going well, how does that play out for somebody who may hit some difficulties during their process?
Yeah, so that's a very good question. So, these projects don't necessarily go through the way that you plan, and sometimes it takes longer than you want it. And then in this difficult pandemic period, everybody is sort of behind their schedule. So if that happens, the first thing you have to do is reach out to us and then explain what's going on. We may or may not be able to provide you with much guidance, but it's better to check with us, and I will do our best to help you. Having said that, these are grant mechanisms, means that once we have your grant funded, then it's up to you to produce outcomes the way you want. You would have to send us final progress report to let us know how much you have accomplished, but it is not an obligation to you have to meet all the goals as you proposed. That is slightly different for the cases of fast track. So like I said earlier, fast track grants have two different portions. So although you get a funding approval for both portions in the beginning of the funding process, but after you finish your phase one work, you have to send us your updated progress report. And based on that, we will go through our own internal review and then decide whether or not we will fund your phase two portion. But otherwise, it's a grant mechanism and there's no dilutive components. So once you have the funding, you can go on and do your own work as best as you can.
All right, we have a question from the audience from Eric Nelson, and it says, can you talk about the U01 grants and what is in scope to apply for U01?
So U01, we have this for our clinical trial cooperative project. So I'm not the best person to give you much details, but basically U01 is much bigger than the clinical trials that our SBIR program can support you with. Having said that, it's about a 5-year project, but it requires a lot more interaction between the applicant or grantee with our clinical trial program directors. So there are constant interactions and communications between the grantee and NEI.
All right, thank you. And seeing no more questions in the chat room, we're and no hands raised at this time. I think we just go ahead and open it up for the general discussion. So I'd ask the team for Odylia, could you please come on camera and join, as well as the other panelists, Dr. Dalkar and Dr. Kelly, if you would please come on, and then we'll just have a general discussion, and just sort of, for lack of a better term, shoot the breeze around this topic. And I'll kick it off with one question to Dr. Dalkar. If you could travel back a couple years, 3 or 4 years, and give yourself some advice to your current self, what would it be? What would you say about this process of entrepreneurship? Would there be any sort of key advice that you would give yourself?
I suppose I would give myself the key advice of being careful about intellectual property and divulgation. And I would also tell myself, don't worry too much.
All right, yeah, but I think between you and Rusty, the intellectual property topic came up a lot. So either to Rusty or Denise, could you speak to that a little bit? Because I think that people often aren't sure when is a good time to really pursue sort of protecting your asset, essentially. And I know that other sort of environments or universities are very good at this. They have very good offices that take care of that sort of thing, or really push them to protect their asset, because it is in the interest of the school or the institution to do so. But could you speak from your perspective of, what kind of data, or when do you really know that you have something that really needs protected?
So I guess I fall into the category of scientists who are being well taken care of by their institution. And so it's kind of a reflex for me before publishing something to seek out with my technology transfer office if we can think about putting some intellectual property, making something out of that before it goes out into the world. So I think every time a study comes to an end where we start being convinced about the mechanism of action of a product we want to develop, or we think we have enough data that we know we have something that has value, prior to publishing it, we usually seek out opportunities to protect it.
Russ, do you have anything to add?
Yeah, I think the intellectual property piece is obviously important. There's an optics when it comes to investors, and most investors want to know that there's freedom to operate and that someone else isn't going to ultimately block you. But it is one strategy that must be considered among other strategies within rare disease. For example, there's regulatory exclusivity in the US and Europe that doesn't have the duration of IP, which is typically 20 plus years from filing. But look, this is difficult space. You take a novel therapeutic such as gene therapy in an ultra rare, you're likely not to have competition, especially if you've already addressed the market. So in those scenarios, you would rely more on regulatory exclusivity, and you should probably be less concerned about IP, but it's still, if you have novel technology and you're going to file not only for methods and for product claims, but for use claims, you might as well go that route, assuming that it doesn't become cost prohibitive to you. But if you're a venture-backed company, that's just part of the diligence criteria. They will perform the IP review for you, and of course, those proceeds of a venture-backed company will pay for that.
Great. And Rusty, real quick, we had a question a while ago that was directed towards you, and it says, if we have a project that could fit within the RD Funds portfolio, what is the best way? Oh, I'm sorry. Go ahead.
I think Eric Nelson just asked the question in chat. about giving 1 or 2 examples of pharma discovery alliances. Should we address this prior or afterwards?
I just want to get to Rusty's question, just his first question, since it was asked so long ago. I just want to make sure that we get that answer for that person. May have to run off, but it's basically what's the best way to contact the RD fund? Just quickly, Rusty, just so.
Yeah, we're very accessible. So the management team, Ben Yerxa, Jason Menzo, Peter Ginsberg, and myself, you can reach out to any of us. And if whomever you reach out to cannot answer your question, we will find answers internally.
Sure. And Eric, would you actually like to go ahead and unmute yourself to ask your question?
Sure. Hi.
There you go.
So I'm a business development specialist, and I've just taken on responsibilities for 2 of the foundation's projects that are at the discovery stage. So I know a lot about drug development, but not specifically about starting retinal disease therapeutic programs and then the track towards pharma alliances. Can you just give examples of 1 or 2 pharma companies, what their profile is regarding discovery, research, what kind of technologies are they looking for? For instance, either Novartis or Bayer, and then which conferences are they going to now in the digital mode?
And Rusty, would you like to take this one?
Towards me, yeah, I'm probably not the best person to answer all those questions, but within our space, you know, OIS is a great platform. So many of the companies in our space, whether it be a startup or the strategics, you know, the big pharma partners are present in these companies. So I would encourage you to start with OIS. In terms of what are the interests of big pharma, it varies rapidly with technology. But I think the Videri exit is a great example of what Novartis was interested in, and that's the field of optogenetics. So Videri and its founders from Berkeley, where Denise spent a few years, developed some optogenetic technology that made cells within the retina, the inner retina, that aren't normally responsive to light. So I think this type of novel technology that can be broadly applied. It's variant agnostic, and it could also be applied later in later stage of disease.
I think maybe it's worth highlighting that Videri had also the technologies for delivering those optogenes, like the optogenetic. So it's the vehicle plus the technology. So I think that also is an attractive feature, maybe.
Yeah, that's a great point. In this gene therapy world, and in this the power of directed evolution to come up with vectors, capsids, serotypes that target specific cells under specific promoters. That's exactly what I think Novartis found attractive. But yeah, I think it's gonna, the field is gonna continue to evolve, and we're always catering to, as you know, Eric, we're always catering to ultimately to big pharma and the public markets. And so, I think it's true for any industry, but our field's changing rapidly. As Denise mentioned earlier, cell and gene is hot right now, cell therapy and gene therapy. So you'll see a lot more activity in that space.
Thank you.
You're welcome.
Dr. Lee, I believe we have a question directed towards you, which is, how as academic scientists, can we have access to those companies who they can partner with for small business grants? And can you give some examples?
Well, that's again a very good question. There are people out there, scientists out there looking for partners, collaborators from academic side, as well as industry side. Industry side, they are looking for academic collaborators, and in academic side, they are looking for industry partner with a special expertise and experience in commercializing our product. So there are many ways to approach to this issue. You can attend these conferences being held to look for investors and commercial companies. We have also a small business conference NIH holds. So if you're an academic investigator who attend these meetings, you'll have a chance to meet those people. In addition to that, we have some publicly available database, including reporters. There, you search for SBIR and STTR, and you will be able to come up with companies who have been successful in securing NIH grants. Then you can reach out to them, and then or you can do a quick Google search of their websites and see if they have something you know, can be integrated with your own projects or work with your project. Then and those are the places you can start from.
All right, thank you for that. I did want to leave some sort of time for the team at Odylia to either jump in with either comments or questions. So Ashley, is there anything that you would like to add or questions you have for the panelists?
Sure, I actually just had a more detailed question on the formatting of the SBIRs, STTRs when you're speaking, Dr. Lee, about the percentage of work being done under each entity. We had previously had a webinar on working with CROs and CMOs, so maybe you can comment about how those are seen within that equation, especially in the late IND stage when you are quite often working with CROs for maybe toxicology studies.
So I have to say that individual cases have to be handled individually. And if a grantee has a question or an applicant has a question, it's best them contact me and then have a private chat. Having said that, for phase 1 and phase 2, we have a different work assignment requirements. So phase 1 SBIRs, we ask for 2/3 work will be done by the SBC. Once it gets to the phase 2 and beyond, then the requirement goes down to 50%. Then you have a 50% of the work that can be outsourced. Now, getting back to CROs and subcontract works, those are very difficult areas, and individual cases made different. If it's something that under your control and it is just a fee for service work that does not necessarily constitute a part of a specific aim, then it can be considered as just the additional purchasing that you're doing. But once the animal study is being operated by the company on their own, then it becomes a difficult area.
Okay, that's helpful. I think that gets to the meat of what I was asking. And Chad, if you don't mind me jumping in with a question for all the panelists. I think Odylia works a lot in the rare disease space with a focus on retinal, but also broader outside of the retina disorders. And one of the challenges we're often coming up against, and it's one of the reasons Odylia was founded, was the commercial paradigm for rare disease. When there's a low prevalence disorder, it makes that commercial the math on that much harder. So I'm curious if the panelists have any commentary on that shifting landscape and the commercial potential for rare disease.
I think rare diseases, if you put all of them together, actually, they account for a lot of disease. So that's with that in mind, I think it's really, they should not be ignored. But in the specific landscape of retinitis pigmentosa, if you look at each mutation, you can consider them very rare. But then maybe the way the field has done to overcome that issue of making a treatment that is going to help so few people has been to focus on treatments that are more, that are converging on the phenotype that is common to all RP. So instead of doing mutation-specific treatments, targeting the common pathway and phenotype and making more things that are focusing on the symptoms has been a way of counteracting that issue. So maybe that's one way of approaching the problem. I don't know if that answers your question, or maybe a
Yeah,
partial.
I'm just curious what the thoughts are of the panelists. I think there's a lot of different ways we try to think about things, and I'm always curious how others in the space are thinking about the future.
So yeah, one way is to focus on mechanisms that are common to few rare diseases, and thereby bring them together in that way. And then second aspect is, once you think about making technologies, there's always more than one application in one disease, and those can be core technologies that can be useful outside of the context of that rare disease, and maybe there is something to leverage out there as well. I don't know if what I said is too abstract. Maybe Rusty can complete with more concrete words.
Yeah, no, I think about some of the transcription factors and specific to the eye, Ashley, that regulate cone rod differentiation. And so depending on where you are in the hierarchy of that pathway, you could affect multiple variants, as Denise mentioned. And I think, another point is, this is something we've talked with you about, Ashley, is how do we reduce costs?
Mm-hmm.
That's, pricing, reimbursement is always going to be an issue. Right now, the price of Luxturna is fairly expensive. But, from everything that we've determined, the cost of vision is also very expensive, right? Or the loss of vision. So how do we reduce cost? And manufacturing certainly has to be in that conversation. It's something that we struggle with greatly, and hopefully we'll figure that one out.
I think maybe as years go by, I mean, if you look at sequencing, for example, the cost of sequencing has gone down so exponentially. I think maybe we can hope for that kind of things to come into the landscape of producing GMP grade vectors that will help lighten up the costs. I mean, I have plenty of hope that the regulatory approval mechanisms are also going to be lightened up a little bit. So as we see more and more of these therapies coming into the clinical application with hopefully results that are as good as they are right now, maybe there's going to be also less costs, because there's going to be less regulatory checkpoints that are going to be costly. And maybe with those two, we can make headway in bringing the costs down and making them available tomorrow.
Yeah, and I'll add that in addition to novel and streamlined clinical design, a component of that is natural history. And that can be very expensive and timely. And so the foundation under Todd Durham's leadership is trying to figure that out. And this is in partnership, obviously, with our SAB, Jackie Duncan, Mark Panisi, and others, but to try to figure out how to take the basket approach to ultra rares. Using Denise's example, is how can you take a number of common RP variants and build a data set that makes sense.
I can
So
say two things on the topic, which is that I've just been doing a lot of commercial assessment work for other types of therapeutics outside of eye disease. But what I found that was surprising was that the insurers in the US are willing to pay a lot for a therapeutic as long as you can prove that it works. So the quality of the clinical data and safety data does need to be quite good, and that needs to be incorporated into the program, and then you can charge quite a bit. From the insurer's point of view, if it's a rare or ultra rare disease, the cost to their overall plan, if they're covering, for instance, 5 million lives, is relatively very small. If it's only going to be on their particular plan, say Aetna, 20 individuals over a certain period of time. So I think the cost for the rares or ultra rares is less of a consideration than does it actually work. And the long-term safety data, they wouldn't want to pay a lot from their plans, and then a year later, the therapeutic blows up because it had an unexpected side effect. The other factor is, do the researchers, say in ophthalmology at the medical center, do they insist on having the therapeutic being covered by their medical center? So at Mass General or Cleveland Clinic, if those clinicians are saying, you know, this is the huge breakthrough, we want to have it, then that will sway the insurance group covering that medical center to cover it.
Yeah, I was going to say pretty much the same line of thoughts. So NIH supports a lot of early stage investigations. We invest a great deal on basic science as well. So you have a R01 mechanism there. You have R24 mechanism for TRP, a little bit closer to translation, but still falls into basic science realm, and then SBIR mechanisms for sort of early stage development for potential commercialization. And then we do have quite a bit of grants fall into those categories, what we call rare disease. So once you have a good data to move on to the regulatory process, I think you will attract a lot of investors out there.
Thank you for that. Ashley, did you have a follow-up?
No, that's it. Thanks,
Oh.
Chad. Go ahead.
No, so a part of this session we want to talk about was sort of the future of ocular drug development. And we have a timely question from the audience. How do you think about the potentials of CRISPR gene editing for retinal disease? And it says this is more of a gene or mutation specific treatment and the target population is even smaller. I can sort of jump in with that just to get my perspective on it. And I'm always a glass is half full type of person. And I really think that as we sort of refine the CRISPR technology, as long as with the other complementary technologies that go along with it, I think that it's really has a big sort of upside for particular retinal diseases, especially the ones that are well identified and their etiology is well known. I think that, you know, CRISPR is going, as long as we can refine it and make sure that it's safe. I think the number one thing to consider with CRISPR is off-target effects and that what is acceptable, what is not. Some of our programs, you know, we're looking at sort of like, well, is on target, you know, 90% of the time and off target 10% of the time okay? Who knows at this point? But I know that it would make me feel very uncomfortable if any part of my genome was being edited it wasn't not intended to because of the adverse effects associated with it. So all in all, what I'll say is that I think there is definitely a huge promise of CRISPR technology for sort of genetic-based diseases of the retina. So I'll let anybody from the panel would like to jump in and sort of give their 2 cents on what they think about CRISPR technology for retinal disease.
Sure. Just as a quick answer, I would say optogenetics is today, CRISPR is tomorrow.
Love it. Rusty or Dr. Lee, would you?
This is Rusty. I'll agree with Denise. I think it's not quite there yet, but it's close. And we have interfaced with a number of the leading experts in this field, and they're very close. And I think that, you know, it's a great point about variants. But in many, including some of the larger IRD markets, there are dominant variants or variants that SNPs, if you will, that are quite frequent. So I think there's room for CRISPR even in the context of the size of the market.
Yeah.
Go ahead.
No, go ahead.
I was just gonna say that we do see CRISPR-based technology applications, and we do fund those as well. Just, you know, when we see those grants, that means it's still 5 to 10 years away from having any commercial product, but still those do come in our realms and our attention.
Yeah, I would agree with what everyone else has said so far. We've evaluated it And I think just we're more interested in the technologies that we know are achievable today. We're not an investment group for future technologies, but I think there's groups out there, there's biotechs that are starting that work now, and willing VCs that are willing to jump in and invest in the future of those technologies. And I think what will be interesting to see is the second gen aspects of the platform kind of evolve over time. And it might not be the first iteration of CRISPR, but it may be the things that follow on, which are starting to look really promising as far as off-target effects. And I think it's also interesting when you start to combine that with regenerative medicine, especially in the eye. So it's not just treating and stabilizing pre-loss of vision, but maybe the potential down the road of actually replacing damaged cells. Some of that is really interesting work. And when you add in the CRISPR aspects, I think there's a lot of potential about 10 years from now, maybe less, hopefully.
I saw that Leslie popped something into the chat room. Leslie, would you like to comment on that, please? Just let our audience know sort of this, I think it was the NIH report that Dr. Lee spoke to. Could you talk a little bit more about that?
Sure. So that's an awesome reference as far as what NIH has funded and the companies that people have partnered with. And there seem to be numerous questions about partnerships, collaborations. And so that's just a really good website to kind of see the trends, I guess, for lack of a better word, and also get an idea of who a good potential collaborator may be.
All right, thank you for that. And I see we have a hand raised, so I believe it's from Hamza. And sorry if I'm not pronouncing your name correctly, but could you please unmute yourself and ask your question? Nope. Okay. Well, never mind. We'll move on. If you do have a follow-up question with that, please just type it into the chat box and we'll be sure to answer. One thing I wanted to mention is I always found it very impressive how it seems that often that the retinal or the eye or vision in general leads the way for emerging technologies. And I know that it's due to a lot of the accessibility of the retina and whatnot, but it's sort of very interesting to work in the field as these emerging technologies sort of roll off from the bench into the preclinical line or even into the clinic, that often the retina and vision lead the way. And so I think it's just a very exciting time to work in this space. But I would like to ask the panel, we have about 5 minutes left of this. Are there any other emerging technologies? And I'm sort of on the wild side of things. As Rusty knows, I often mention from my past experiences of working at DARPA that I'm always interested in sort of the wild ideas when it comes to technology development. Are there anything from the group that they're really interested in, that they've seen at the conference or seen in a sci-fi film even, that really sort of piqued your interest when it comes to technology development for the retina and for restoring vision?
Well, I can start off. We see the full spectrum of technologies come through. We take them all seriously, and we're certainly fascinated by even the classical approaches of neuroprotection and the pharmacological intervention. But in addition to gene therapy, which gets a lot of press, optogenetics, of course, and even gene editing, there are a number of RNA therapies that are coming forth that are very exciting. In fact, in the last 6 months, I've learned of 2 or 3 new types of RNA therapies that have just been recently discovered over the last couple of years. So I think it's the genetics piece of this is at the forefront, right? Our ability to understand not just a variant of an IRD, but what else is at stake? What other modifiers, what other gene modifiers are playing a role in disease and disease progression? It's very exciting.
Okay, I cannot get into any specific applications or projects, but so the group is much more in tune to retinal disease and biologics and gene therapy. But from NIH, SBIR programs, AI is a big thing right now. So a lot of people are developing tools using AI. So from the device point of view, using AI to analyze data quickly and go back to the patient. So that's one of the things. And then even from genetics and data analysis side, AI is being used a lot. So big data, how to analyze data the way you want, will become a big hot topic in near future, I would say.
All right, I see we have someone in the audience that has their hand raised. Could you please unmute yourself and introduce yourself and ask your question?
Hi, do you have my voice? Can you hear me?
Yes, we can hear you.
Okay. My name is Mahnaz. I'm actually in this conference and I enjoyed it a lot. I'm from Tehran, Iran. And one thing was very interesting for me regarding your question. I think Professor Lee somehow mentioned it. When I started, my background is in chemical engineering, polymer engineering. I actually studied multidisciplinary. And when I got back to Iran 10 years ago from United States, I started my research in biomedical engineering on AMD and retina tissue. And what happened was I started from just, treatment, drug delivery systems and designing bioreactors in order to test new actually drugs for retina tissue and how we can regenerate even retina tissue. But beside that, I mean, it's not a question. I just, when you ask, what do you see in future? That was one thing that at least I felt, and I thought it would, maybe you like to hear about it as well. But it came to my mind, I started from very simple, just a disease. And when I started learning more and more, I came to a point because now even I'm working on neural tissue, I was thinking, how can I create a screen from retina tissue, from photoreceptors, and get even the signals to neurons. Then I work now in neural tissue as well. And last few months, my mind were busy and I was thinking how it will happen if one day we can make a screen totally from retina tissue, and it would be totally a biological screen. And what will happen if in future, even in the space, we make, some satellite that the screens and everything becomes a biological totally, that was when you asked about, the retinal, because all I had in mind from my research that I do simulation work, I do, experimental work, little by little, I came to the point that I wanted to make a big retinal, tissue and create a screen and create image from that. And later it came that why not we can use it in our, TVs and, maybe it is the next generation of our TVs. I know it is very crazy, it is very far, but I don't know, it sounds very actually interesting to me. I don't know.
Yeah, well,
Maybe
thank you for
like
sharing. We really appreciate that. And no, that's very interesting. So yeah, when we ask questions like that, I think nothing's off the table. So please don't feel like it's crazy or anything. It's actually just the natural evolution of creative minds thinking very far outside the box. We are actually at time, so I don't want to keep any of the attendees or panelists any longer. I know that you have other commitments, but I would like to thank the panelists so much for your time and for the information that you've shared. It's greatly appreciated. I want to let the audience know as well. Again, like the others, this webinar will be available in probably within the next 5 days or so on the Foundation Fighting Blindness's YouTube page for the reference. Please share it with your friends and your networks. If you have any questions, please feel free to reach out to everyone. Ashley, would you like to say anything in closing?
We just appreciate everyone's time, all of our speakers and all of the participants for taking time out of your day over the past 4 webinars to participate. And thank you, FFB, for the great partnership. We look forward to exploring it further. Thank you.
Us as well. All right, everyone, take care and have a great rest of your day.
Bye.
Bye.
Thank you.